Dr. Burgess from the world famous Jackson Laboratory. I will let you read the rest of his bio on the speaker's page. His bio speaks for itself. But what I would like to tell you about Dr. Burgess is that in addition to being brilliant, he is incredibly generous. When we first asked Dr. Burgess to help us with this convention, it was to serve on the scientific program committee. To my delight and a little surprise, he said yes. He was instrumental in putting yesterday's researcher meeting together. And then we decided that one of the themes yesterday, the program committee did, should be mechanisms and models of disease. And the rest of the program committee told me that Dr. Burgess would be the best person actually to lead a session on that topic. It's highly unusual to ask a program committee member to take on that kind of role, but ask we did and accept he did, again, to my surprise and delight. And if that was not enough, when we decided to have this session on academic research today, you guessed it, we asked, he agreed. Frankly, he may stop taking my calls. Dr. Burgess, please take it away.
Yeah, thanks very much for the invitation. And maybe I'm a sucker, but I'm also excited to be part of this program and part of the CMT research effort. So I actually appreciate the opportunity to contribute. So as Keith mentioned, I am a researcher at the Jackson Laboratory. This is a picture of the Jackson Laboratory. It's in Bar Harbor, Maine. Not a bad place to live. And the lab is pictured here in the foreground, and Acadia National Park is pictured here in the background. But we're a nonprofit research institute. I run a grant-funded laboratory there, primarily NIH funded. We've gotten other funding from foundations like the Muscular Dystrophy Association as well, but very much a basic science institute. Similar to the research that would be done at a medical school, basic science department, a genetics department, or a neuroscience department. And we also have a very strong effort in preclinical studies, and we have a large group at Jackson that we specialize in mouse models of human diseases, and including neuromuscular diseases and Charcot-Marie-Tooth disease. And so as drug companies develop compounds and want to test them in proof of concept studies, we have a large group that actually helps with that sort of process also. We're sort of the experts in analyzing mouse models of human disease. So that's sort of my current role and my background here. But what I want to tell you about today is a little bit about what's going on in basic science. And actually, I really appreciate following Will. He came to my lab and worked there over 10 years ago now. But it's been a pleasure interacting with him on a regular basis since then. But the basic science, and Will set this up, I think, really nicely. The basic science that labs like mine do really set the foundation for the translational research and the drug development that comes next. So as Will was talking about the phases of drug development, phase 1, phase 2, phase 3, we're essentially phase 0. We're sort of the identify the target and do the proof of concept studies that are going to be necessary to then launch into those more complicated studies that start involving human subjects. But the basic science is really foundational for doing that work, quite simply because you really can't cure a disease that you don't understand. You're kind of flailing in the dark until you really start to understand the cellular and molecular and genetic mechanisms that you need to target to improve things. So that's really, I feel, my role in CMT research is largely to identify these targets, and then, as Will described, hand this off to biotech and pharma companies to actually develop the drugs, and to clinical research teams to actually work with the patients and do the trials. So there are kind of 3 areas that I'd like to cover today, and some of that is some of the progress that's been made in gene identification for Charcot-Marie-Tooth disease. And then also, what do all these genes tell us? So we now know a lot of genes are associated with CMT. Is that a good thing or a bad thing? And what are we doing with that knowledge? What are the challenges and opportunities that presents? And then I also do want to touch on a few, generic, if you will, strategies. And this actually builds nicely on Susan's question for Will. In some cases, I think we're going to need very targeted therapies where it's going to be very specific for a single subtype or a small subset of Chikomori-tooth patients. In other cases, we might try to do something that's going to be generally beneficial in lots of patients. And there are a few of those on the horizon that have come out of the basic science, and I want to touch on those today also. So, going through this in terms of gene identification, when I first started working on Chikomori tooth disease, which was in the early 2000s, that was the major focus of the research. It was really human genetics trying to identify the genes that were involved. And at this point, it's safe to say there are at least 100 different genes, 100 different mutational sites in the human genome that are associated with Chikomori tooth disease. And in terms of the actual identification of the causal mutations in those genes, we're doing pretty well for demyelinating neuropathies. Depends a little bit on who you ask, but there's probably around a 90% chance of a genetic diagnosis for a demyelinating form of Chikomori tooth disease. We're doing less well for the axonal forms, where there's probably still less than a 50% chance of a genetic diagnosis for axonal CMTs. So this is I think an opportunity and also a challenge. We know there are 100 different forms of CMT. Does this mean we have 100 unique diseases? At some level, yes, but there may actually be a blessing in disguise there. I'd be interested to have Will's opinion on this, but I think one of the challenges facing some other diseases, similar diseases like Alzheimer's disease or Lou Gehrig's disease, ALS, is that everyone acknowledges that ALS is not one disease, it's not one monolithic problem. But we don't know, the genetics is not as clean, the subtypes are not as well defined. And as a result, we don't really know how many different diseases ALS actually is. And as a result, when you try to develop a therapy and you go into clinical trials, the patients all get lumped together as ALS patients. And it could be that there's some real benefit for a real subset of those patients, but that signal gets lost in the noise because maybe 95% of the patients don't respond at all. So the fact that we actually have the subtypes of CMT well defined, and we can start to address these things. Is this going to be a general, general application to CMT, or is it going to be a very specific, therapeutic for CMT? Is probably actually an advantage because we can I think a little more clearly about what we're trying to do. That said, 100 unique diseases, there are going to be common mechanisms and common pathways in these different 100 different genes, and I'll touch on that a little bit. So, maybe we're not looking at 100 bespoke therapies, but we could maybe break that down to a few dozen or something like that. It might not be quite as daunting as it seems. Also, in terms of the gene identification, this really gives us the opportunity for these bespoke treatments. And Will touched on gene therapies and antisense oligonucleotides that are really directly targeting the genetic change, the mutation that's causing the peripheral neuropathy. You obviously have to know the gene and know the mutation, know that DNA sequence before you can design these gene therapies and ASO strategies. So the gene identification is absolutely critical for that sort of genetic medicine. And then also for gene-specific drugs, and maybe not really gene-specific, but CMT subtype-specific drugs. And again, knowing the genes that are responsible for these different forms of CMT helps us kind of categorize these things. Maybe not 100, but maybe a couple dozen different CMT types that we can be thinking about. More specifically than just demyelinating and axonal. So in terms of generic strategies, and I'll touch on a couple of these in more detail in a moment, there are a few things that are out there that are kind of interesting. And one, we heard a great talk yesterday from Zarif Sahenk, who's at Nationwide Children's Hospital in Columbus, Ohio. And she's using neurotrophic agents delivered through gene therapy to both improve the health of the nerves, but also to promote nerve regeneration. And this is a really interesting approach that I think has potential to treat lots of different forms of CMT and maybe even other neuromuscular diseases like ALS or spinal muscular atrophy. So, this is a kind of an exciting possibility. And then two that I'll mention in more detail in just a moment are inhibitors of a protein called SARM1 and SARM1 actually promotes axon degeneration in people. So inhibiting SARM1 presumably will prevent axon degeneration. And then a second class called HDAC6 inhibitors. And HDAC6 is histone deacetylase 6. But in neurons, it targets a protein in the axon called tubulin. And acetylated tubulin helps things. Tubulin is basically the railroad tracks of the axon. And acetylated tubulin is essentially greasing the tracks, and things work really well. And deacetylated tubulin and HDAC6 deacetylates tubulin. Deacetylated tubulin kind of slows things down and prevents axonal transport on these microtubule tracks. So things that inhibit HDAC6 are going to promote tubulin acetylation and therefore promote axon transport and axon health. And that's being explored in several different forms of CMT also. And I'll talk about that a little bit more in a moment. So to just go through a few examples then of how we've gone from gene discovery to mechanism to possible therapeutic, I just want to list a couple of things that I think have been exciting in the recent past. And the first of these is a paper that was published by a large consortium of people, headed by Stefan Zuckner and Andrea Cortis, who was working in his lab at the time. And it identified recessive mutations in a gene called SORD, S-O-R-D, as a common form of axonal neuropathy. And this was interesting because it actually turns out to be, quite a few of these patients. The genetics was a little tricky here for some technical reasons, but once it was identified, they realized like there are actually quite a few of these patients out there. And the reason this is exciting, and the reason the title of this paper says a potentially treatable hereditary neuropathy, is that we know what SORD does. So SORD is involved in sugar metabolism. So you go from glucose to sorbitol and sorbitol to fructose. And SORD is sorbitol dehydrogenase. So it's the enzyme that catalyzes this change from sorbitol to fructose. As a result, when you get rid of this enzyme, when you kill SORD activity, you have a buildup of sorbitol, and that seems to be toxic to the axons, toxic to the nerves. So the other way that you could prevent the buildup of sorbitol is by inhibiting this enzyme, aldose reductase, right? So you don't even get to the sorbitol to have it accumulate. And the good news about this is that aldose reductase inhibitors are actually out there. In fact, they're approved for clinical use in Asia and in Europe. They're just not approved in the US, but trials are in the planning stages and possibly already underway to start looking at the use of aldose reductase inhibitors in this specific form of Chikomori tooth disease. So here's an example where I think the gene identification sort of led to the mechanism, and the mechanism, since we understood it pretty well, actually very quickly led to a possible therapeutic. In work from my own lab that Will mentioned, and I'm thrilled with the attention and the excitement that this paper is generating, we identified that dominant mutations in tRNA synthetase genes activate this cell stress pathway called the integrated stress response. And the integrated stress response is kind of like an emergency brake for your cells. So if they're starved for protein, or if they're infected by a virus, they'll activate this integrated stress response, kind of shut everything down until that stress passes, and then the cells kind of kick back into gear. And we think that what's happening with these tRNA synthetase associated neuropathies, and there are 5 or 6 different tRNA synthetase genes that all cause forms of Chikomori tooth disease. We've mostly studied 2D caused by GARS, but we are studying 2 or 3 of the others also. We think that essentially what's happening is there's a problem in protein synthesis, and then the cell's response to that is to further shut down protein synthesis. So to use the car analogy again, your brakes are sticky, and the car's response to that is to throw on the emergency brake, right? So you've got a problem, and the cell's response to the problem actually makes the problem worse. And the good news about this is this GCN2 protein is the sensor that turns on this integrated stress response. And we showed both genetically and using an experimental drug that inhibiting GCN2 actually greatly improves the phenotype and lessens the severity of the neuropathy in these mice. So we're actively now pursuing this as a drug development program. Again, that isn't work that would be done in my lab, but we're talking to pharmas and biotechs and trying to get people to pick up this GCN2 inhibitor idea as an indication for these tRNA synthetase associated neuropathies. So again, this was just basic science research. We were trying to understand how these genes cause this disease. And in the process, we figured out they were activating the integrated stress response. And in the process of testing that, we figured out that it was actually a therapeutic target. To spend just 1 minute on how complicated this can be, later today, you'll hear from Maurizio D'Antonio, and he's also studying the integrated stress response, but in the opposite direction. So in forms of Chikomori-tooth 1B, for example, the integrated stress response is a good thing, and you actually want to keep it on. So the cells are kind of out of control with misfolded protein in that mutation that causes CMT1B, and in that case, keeping things kind of damped down, keeping the emergency brake on is a good thing. So in some cases of CMT, you may want to inhibit it, and in other cases of CMT, you may want to keep it activated. So just quickly to discuss a couple of these more generic approaches, work that was actually started in Drosophila, fruit flies, identified this really interesting protein called SARM1. And SARM1, it turns out to be necessary for injury-induced axon degeneration. So if you crush an axon, if you cut a nerve, this might happen in an accident or an injury. SARM1 is part of the process that basically causes the nerve distal to that injury to degenerate, and that clears the way for the nerve to then regenerate successfully. But SARM1 is a little bit like walking around with a loaded gun, right? You've now got this protein that's geared towards axon degeneration, and it's becoming increasingly a question whether this could be a therapeutic target in forms of Chikomori-tooth disease. And that's something my lab is exploring, and it's something that lots of other labs are actively investigating also. But on the encouraging side, inhibiting SARM1 in various ways, and right now there are not drugs available to do this, but there are several companies that are actively developing these drugs. But SARM1 inhibitors are effective at preventing diabetic neuropathy. They're effective at preventing chemotherapy-induced neuropathy. So I think there's some real potential in Chikomori-tooth disease as well. We just need to figure out which forms of CMT is this going to apply to. I would love it if it was all of them. It probably won't be all of them, but it might be several of them. So that's an area of active investigation in my lab and in lots of other labs. And then the final kind of generic approach that I want to mention here is this use of HDAC6 inhibitors to promote axonal function and axonal transport, like I mentioned. And this was first reported in a mouse model of Chikomori-tooth type 2F, and that was work done by Vincent Timmerman and Ludo van den Bosch in Belgium. And they showed that these drugs that inhibit HDAC6 really improved the phenotype, decreased the neuropathy in these mouse models of CMT2F. Subsequently, a couple of papers have been published. I'm on this paper and another paper from Ludo van den Bosch showing that this also works in Chikomori-tooth 2D mouse models carrying GARS mutations. And then recently, it's also been published as being possibly effective in Chikomori-tooth 2A mouse models with mutations in mitofusin. So, this looks like it might be, a generalizable approach that's going to help lots of different forms of CMT. I think a caveat to this is the effect size here is not overwhelming. You will not take an HDAC6 inhibitor and be good as new. And I think it's kind of back to what Will was talking about. You're going to need to address the primary genetic mutation. You're going to need regenerative therapies. Something like an HDAC6 inhibitor might also provide a little bit additional benefit, of slowing down or preventing some of the axon degeneration in the first place. So probably not what you would truly call a cure unto itself, but there may be reason to expect some benefit in at least some forms of CMT here. And again, this is being actively explored using mouse models of the disease and other experimental model systems, but is rapidly moving towards clinical trials in a couple of different companies also. So to just give you a quick summary, and then I'd be happy to answer a few questions. In terms of the gene discovery, which for the last couple of decades has really been the focus of CMT, we've really made excellent progress. There's still some work to do, especially for the axonal neuropathies. This is obviously, really beneficial for just patient diagnosis. You can actually genetically define your subtype of CMT, but it's also essential if we're going to do things like gene therapies and genetic medicine. You've got to know the mutated gene. I think now that we're at 100 genes associated with CMT, there's a little less emphasis on the gene discovery and more emphasis on identifying the pathways and trying to identify therapeutic targets. And I gave you 2 examples of that with the SORD mutations and using aldose reductase inhibitors, and with the GARS mutations that cause Chikomori-tooth 2D and using GCN2 inhibitors. But it's actively underway for lots of forms of CMT, and the work from Maurizio D'Antonio, for example, to keep the integrated stress response active. There are drugs that can do that as well. And then in terms of these general strategies, I think SARM1 inhibitors are an exciting area to investigate for CMT, and that's in its fairly early stages, but is really underway in a lot of different labs around the country, including mine. And again, these general strategies to improve axonal function and axonal transport, like HDAC6 inhibitors and neurotrophin gene therapy to promote regeneration as possible, broadly applicable therapeutics are in the testing phases as well. So that's, I believe, all the slides that I have. And with that, I'd be happy to stop sharing and answer any questions that may have come up.
All right, fantastic. Thank you very much, Dr. Burgess. We do have time for maybe just a couple of questions here. So I think one is quite specific from the Q&A, and you spoke about SARM1 inhibitors, and I think you briefly said we're not sure yet which kinds of CMT they may or may not work for. That's an active area of investigation, but you might be able to speak to this question, which is, are SARM1 inhibitors potentially applicable to CMT1, and in particular type 1B?
Yeah, that's a good question. I guess my answer right now is I hope so. The form, when you have a demyelinating neuropathy, there's still some axonal dysfunction and often some axonal loss, even if it's secondary to the demyelination. And I think one of the areas where that's been, I think, well established is CMT1X, GJB1 mutations, but it may be true in some forms of CMT1B also. So I think, maybe logically SARM1 inhibitors are maybe best suited to the axonal neuropathies, but I don't think I would want to rule them out for some of the demyelinating neuropathies as well.
Yeah, and of course, ultimately we'll know when we see the data, right? I mean, that's what we have to do.
Yeah, we'll know when we test it. That is true.
Yeah. This is a great question here that I'd love to pose to you, and this will be our last question for this session. From Robin Schultz, what is the challenge for the rare forms of CMT? And I think she really means ultra rare, fewer than 100 patients. What do you see as the main challenges there?
Oh, and hello, Robin. That's a great question. One of the challenges is just engagement, right? And it's not that I wouldn't want to work on every form of CMT out there. It's just that everyone just has only so much bandwidth. And obviously, the more patients, the more attention and the more funding that these areas get. I think one of the advantages perhaps for these ultra rare diseases is that the gene therapy strategy actually is really, I think, primed to target those ultra rare diseases. And it's, at least in theory, a little bit easier to pipeline. If we know the gene that's mutated, we know how to fix it. We can put it in an AAV. We can put the gene back. There are always details, how do we deliver it? When do we deliver it? But conceptually, that's quite straightforward. And at least where we stand right now, and Will kind of mentioned this, one of the challenges with gene therapy right now is actually manufacturing the gene therapy vectors. So we do not have the manufacturing capacity to treat all the Duchenne's muscular dystrophy patients out there, even if the gene therapies that are being developed prove effective. That's not true for 100 patients. So in that regard, I think this emphasis on genetic medicine and gene therapy approaches actually is really going to serve that community well.
Great. Thank you very much, Dr. Burgess. We really appreciate your time today and yesterday and all the time leading up to the convention. Thank you. Thank
All right.
you.
Thank you very much, Keith.
All right. Take care. Bye.